Good afternoon, viewers. We welcome you all to this video. Today, I would like to give you a snapshot of in vitro contamination, which is a very important factor uh, for the survival of any tissue culture lab and on the way to successful commercialization of any tissue culture lab or any plant, any crop. Contamination, the in vitro contamination especially, is the major obstacle on the way to successful tissue culture. So far, we have seen and we have experienced so far the different kinds of endogenous bacteria like E. coli, Salmonella, and different bacillus types of bacteria and fungi like Penicillium, like Mucor, Rhizopus, different types of fungal infection. It's very common in almost all tissue culture lab. Today I would like to focus on in vitro contamination of Jarbera because this is very vital, very vital. You know why? Because the Jarbera plants, if you see the ex vitro condition, any explant preparation, you will find hundred thousands of unicellular hairs are present on the body of the leaf petiole, on the leaf, on the flower stalk, on capitulum. You will find hundred thousands of unicellular hair, and that unicellular hair might contain the endogenous bacteria. So, even if you sterilize the plant with mercury chloride or any other sterilant, it might not clean the body for different reasons. So that is why, and ultimately everything will be just perish in your tissue culture lab. So very vital you have to be very careful while you're preparing your explant during a sterilization and autoclaving the media, autoclaving the sterile water. Everything it depends on. The success depends on in vitro contamination. So I would now, whatever you have seen, this is pure plant, no contamination whatsoever of any bacteria or fungi. It's a very clean gene bank we have developed so far. But today I will show you the contamination, the bacterial contamination. It is very difficult to get rid of any bacteria, either endogenous or even superficial in the media or in any part of the plants or even the forceps or the any. Even if you work in the laminar airflow, this laminar airflow, it does not necessarily mean that you are 100% safe. Every time you should be very careful of contamination. See, this is it's not early stage, but this is one of the sign of in vitro contamination. This is the bacterial contamination. The color is, you see, the off-white to light yellowish color. You cannot help this culture, except you can have a chance for in vitro rescue, but that will not give you 100% success. Even if you treat the plants with antibiotics or you whatever. So this is very vital. Another vessel, you can see this one. See, is full of bacterial contamination everywhere, the bacteria, but the culture is still good. You see the leaf are still green, but nothing can be done with this culture. We will make a trial if we can get clean the explant and re subculture, but I am in doubt whether we will be able to get rid of contamination here. As many as 15 small plants, if you go through the rescue uh, culture, you might get hardly 2 or 3 plants maximum. Other will not, they will die because these plants are very small and they will not be able to resist the um, early effect of the sterilization procedure. So that is very vital. So you must be very careful in order to save your plants from in vitro contamination. So there are a lot of things that you have to consider if you want to save your plants from bacterial or fungal contamination. There is no chance of viral contamination, but the bacteria and the virus, they are the major obstacle for in vitro contamination. So we will speak later on for other in other videos. We will make a series of video for you to show you the ultimate how we can get rid of, what are the chances, what are the process of get rid of this in vitro contamination. So until next video, have a nice day and keep away from fungus and bacteria. Keep your plants safe. Bye bye.